Hello guys, welcome to my channel Easy Life Science. Here I will be talking about human fertilization and embryonic development, and will be discuss about the structure of human sperm, human egg, and fertilization process, also the embryonic development. So please subscribe my channel Easy Life Science, like my video, share it to your friends, and don't forget to press the bell icon so you'll get the notification of my newly uploaded video. This lecture will be divided into the two parts. The first part is a fertilization and the next one is for embryonic development. So let's start from the fertilization. The fertilization and implantation are the most critical events in the reproduction process. Fertilization is natural process which is carried out by fusion of male and female gametes, which results in a formation of zygote. Actually, in the fusion, male gamete is sperm cell which donates its nucleus, that is a male pronucleus, into the cytoplasm of female gamete, that is egg cell, and in egg cell it fuses with the female egg pronucleus. By fusion of these two pronuclei, zygote is formed inside the egg cell cytoplasm. In human being, the process of fertilization takes place in a fallopian tubes of female reproductive organ, that is vagina. During fertilization, semen comprising thousands of sperms are inseminated into female vagina during a coitus or a copulation. The sperm moves towards the uterus and reach the opening of fallopian tubes. Only few sperms will succeed in reaching the opening of fallopian tube. The secondary oocyte released from the matured Graafian follicles of the ovary and enters into a fallopian tube, where it is fertilized within 24 hours, after which is released from the ovary. If you look at this diagram, here there is a structure of the female reproductive system or organ, and this is a fallopian tube where the matured sperm cells and eggs will get fused. This is a body of a uterus. This is a ovary. Two ovaries are there. This is a vagina. Fertilization in a mammals occurs in a oviduct, and the ova viable for approximate 24 hours after a process of ovulation. Though surrounded by several sperms, the oocyte is fertilized by a single sperm. During meiosis second, the sperm enters the secondary oocyte and completes a meiosis. After this, the secondary oocyte is known as an egg. Both sperm and egg show their fertility only for a limited period. Sperm is alive for 48 to 72 hours in a female reproductive system, whereas the egg can be fertilized for 24 hours before it is released. So this is the introduction for fertilization. Then the structure of a sperm. The sperm cell is a male reproductive cell or a gamete. Human produces a motile sperm with a tail known as a flagella. This part is a tail or a flagella. Human produces sperm cells during a process known as a spermatogenesis. The spermatogenesis takes place in a seminiferous tubules of the testes in a male. Human sperm cell is haploid, so it represents a n number of chromosomes. So that its chromosome number is 23 chromosomes are there in a sperm cells, and these chromosomes can join or can fuse with 23 chromosomes of egg of a females to form diploid, that is a 2n zygote. and the zygote has 46 paired chromosomes. The head of sperm contains chromosomes which when combined with the egg's chromosome which will create a new individual. The tip of sperm head is called a acrosome. If you look at to the tip of the head, this is a tip of head and this is known as a acrosome. The acrosome of a sperm cell is derived from a Golgi bodies. Then the middle piece contains the mitochondria which supplies the energy to move the tail. And this is the structure of human sperm. The next structure of the egg cell or a ova or a oocyte. It is also known as a female gamete. Single cell is released from the ovary of a female which is capable of developing into a new organism when fertilized or united with a sperm cell. Human female produces non-motile egg cell. The ovary produces and stores and releases the egg cell into a fallopian tubes of a female reproductive system through the process known as ovulation. The egg cell contains the nucleus, cytoplasm, zona pellucida and corona radiata. Zona pellucida is a protective membrane which covers the cell membrane, while corona radiata is a protective layer which surrounds the zona pellucida. The sperm adheres to the ovum at zona pellucida during a fertilization. Have a look and see in a detail. This is a structure of a oocyte or a egg cell or a ova. This is a haploid nucleus of a egg cell. Contains one copy of each chromosome, so we can represent it as a N. The mitochondria are there which produces a ATP and this be utilized for a energy currency which is having a DNA known as a mitochondrial DNA which gets passed to the offspring. Then one layer is there and the layer of some granules known as cortical granules, and these cortical granules release the enzyme known as cortical enzymes during a fertilization, and these enzymes hardens the zona pellucida, and because of a hardening of a zona pellucida it inhibits polyspermy after a entry of a sperm cell into a ovum. Then another layer is there known as a corona radiata or a layer of follicular cells, and this layer of a corona radiata protects the inner layer of a ovum and also helps to prevent a polyspermy. This is a nucleus of a corona radiata. Then the zona pellucida, it is a jelly coat which contains glycoproteins, and these glycoproteins protect the inner contents of the ovum until the acrosome reaction of fertilization. It also helps to prevent a polyspermy. So this is about the egg cell. Then the mechanism of fertilization. The fertilization process in humans takes place in several stages involving both the chemical and physical events. Different stages of fertilization in humans are as encounter of spermatozoa and ova, capacitation and contact, acrosome reaction and penetration, fusion of sperm with the egg, and the last step is activation of ovum. So these are the some steps. The first step, that is encounter of spermatozoa and ova. During the fertile phase, millions of sperms travels from vagina to the uterus into the fallopian tube and through the chemotaxis. The chemotaxis means the human vagina produces some chemical substance and this attracts the sperms. So this is known as a chemotaxis. In general, interaction is through facial devices or particular forms of behavior. there is a need of fluid which will be released by the activity of a egg. There are the two types of the fertilizations. It may be external or internal. The external, the external fertilization occurs outside the body of a female. Here, the female lay out the eggs, and outside the body, the eggs are fertilized. In this external fertilization, increased number of eggs produced to ensure the survival of the species, because the female in this case laying the eggs outside the body, and so the eggs are in a danger. So there is a need to produce a number of eggs to ensure the survival of their species. Such kind of external fertilization occurs in a fish and amphibians. Then the internal fertilization occurs inside the body of the female. Here, the ovary produces an egg inside the reproductive system, and that eggs get fertilized through the sperms. Fewer numbers of eggs are produced. In this case, or in internal fertilization, there is increased parental care which ensures the species survival. It happens in mammals, reptiles, birds. Several thousands of sperm reach the egg, and only one egg gets fertilized by a sperm. When the sperm fuses with the egg, it initiates a series of chemical changes that prevents any other sperm from entering, and this is known as a prevention of polyspermy. Then the second event, or the second stage, that is a capacitation and a contact. In a capacitation, the sperm cells after entry into a female reproductive system, it get modified or matured, and it's somewhat known as a capacitation, and this occurs in a female's vagina. Capacitation of sperm occurs in a female's vagina. The vaginal secretion causes a molecular change in a sperm, and which removes decapacitating factors, and so that the capacitation of a sperm occurs, and it takes 5 to 4 hours in case of a human beings. Then the third stage, acrosome reaction and penetration. When the acrosome reaction occurs, a number of proteolytic enzymes are exposed or released by a egg cell. The sperm undergo a acrosomal reaction and release certain chemicals known as lysines and proteolytic enzymes. The proteolytic enzymes and lysines degrades the zona pellucida of secondary oocyte. The sperm fuses its plasma membrane to the oocyte, and contents of a sperm released into a cytoplasm of oocyte. When the plasma membrane of sperm binds with that of the secondary oocyte, the plasma membrane of oocyte depolarizes, and this depolarization of plasma membrane of oocyte prevents a polyspermy. The calcium ions plays a significant role in acrosomal reaction. The main factor essential for acrosomal reactions are optimum pH, optimum temperature, calcium and magnesium ion concentration. Then the cortical reaction soon after the fusion of plasma membrane of the oocyte to that of the plasma membrane of sperm, the oocyte shows a cortical reaction. The cortical granules found under the plasma membrane of oocyte and releases cortical enzymes between zona pellucida and a plasma membrane. Zona pellucida is hardened by a critical cortical enzymes that prevent a polyspermy. So this is a acrosome reaction. To the tip of head of the sperm, this is a head of sperm, middle piece and tail. To the head, there is some cone-like projection known as a acrosome, and this acrosome, after binding to the plasma membrane of egg cell, it releases out some enzymes known as lysins and proteolytic enzymes. And these enzymes break down or degrades a zona pellucida, that is a jelly coat of the egg cell, so that the entry of sperm cell into a new cytoplasm of egg cell is happen. Then about the zona pellucida, it is a jelly coat or it is a covering around a egg, and it is made up of three glycoproteins known as ZDP1, ZDP2 and ZDP3. ZDP stands for a zona pellucida. These are the three glycoproteins. Repeating subunits of ZDP2 and ZDP3 forms a filaments that bound together by a ZDP1. Fusion of sperm with the egg. After the sperm entry, that is sperm release its nucleus into the egg cell cytoplasm, and to the egg nuclei, sperm nuclei get fused. The male nucleus enters the egg cytoplasm and becomes a male pronuclei. As a result of a sperm fusing with the egg plasma lemma, the oocyte nucleus, which is at a metaphase of a second meiotic division, completes that division and giving rise to another polar body. Following the second meiotic division, nucleus of a ovum becomes the female pronucleus. The female pronucleus and entered sperm pronucleus or a male pronucleus get fused to each other. Both of them are a haploid. They attract to each other, meets and fuse, forms a diploid nucleus of a zygote. The zygote is now proceed to undergo a cleavage. Then activation of ovum, the entry of a sperm into an egg triggers the metabolism of the egg for the zygote. Consequently, the protein synthesis, cellular respiration also get increased to the egg cells. Then the implantation, once the fertilization happens, the cells start to divide and multiply within 24 hours in the fallopian tubes. This detaches multi-layered structure, that is a zygote, after 3 to 4 days, and this zygote travels to the uterus, and now we will call it as an embryo. The embryo develops and undergo various stages and get attached to the endometrial layer of the uterus, and this process of attachment of embryo to the endometrial layer is known as implantation. The events which characterize to egg activation are release of calcium. The calcium ions are stored into a endoplasmic reticulum of egg cells, and these are important for the cortical reaction. The second event, cortical reaction, the cortical granules get ruptured, which concurrently release calcium ions and some enzymes. Because of this enzyme, it hardens a Fertilization membrane or a zona pellucida, because of which or because of hardening of these layers, there is no polyspermy. In many species, an influx of sodium ions into the egg cytoplasm that causes a change in membrane potential. Because of an influx of sodium ion into a egg cell cytoplasm, it changes membrane potential, and it is known as a fast blocking of polyspermy. In many species, reorganization of the egg cell cytoplasm also occurs. In most cases, completion of meiosis by the egg, an efflux of H plus ions or hydrogen ions which cause increasing cytoplasmic pH of egg cell, and this activates previously inhibited synthetic pathways of egg cell. It also leads to increase in a metabolism and protein synthesis, which is favorable for zygote for its development. Now about the reactions which occurs in a fusion or a fertilization. Starting from the first point, here it is once again a diagram or a image of the sperm. It has a head. In a head, yellow colored nucleus of sperm is there. To the tip of head there is a acrosome. To the middle, mitochondria, and tail is there. This is a structure of a sperm. The sperm approaches to the egg. Then second event, the sperm having a acrosomal enzyme, and that enzyme digests the egg's jelly coat, that is a zona pellucida. Third step, the proteins on a sperm get bind to the receptor which are present on the egg cell. The plasma membrane of a sperm and egg gets fused. After fusing of these two plasma membranes, the sperm donates or release only the nucleus. Here we can see for the yellow colored nucleus which is of the sperm, it gets released from the head of sperm to the cytoplasm of egg cell. Here it acts as a pronucleus. Already there is a egg pronucleus which is in a blue color, and these two nucleus, female pronucleus and male pronucleus, get fused and forms a zygote which is diploid in nature, and zygote becomes an embryo. Now about a sex determination, we know that father or a male determines the sex of a child. That is, if sperm carries a Y chromosome, the child will be the male baby, or if the sperm carries X chromosome, child will be the girl or a female baby. Now what would happen if the fertilization does not occur? If the fertilization does not occur, there will be a no zygote formation, and egg will degenerate and shed through a uterine lining. Typically we calls it as a menstrual cycle. As early as 12 hours after fertilization, you can see two bundles of genetic material, or we can see for two pronuclei, female pronuclei and male pronuclei, which is from each parent. By 80 to 20 hours after fertilization, these pronuclei fuse and start to become as a one and forms a zygote. Then the next part of our lecture, that is embryonic development. A multicellular organism in early stages of development is known as embryo. In this slide, we can see for 4 cell stage of embryo and 8 cell stage of embryo. The beginning developmental process are always same in all animals. It started with a cleavage, growth, and differentiation. The formed embryo, and after implantation, after fertilization, the diploid zygote undergo a cleavage and a division in a oviduct. The cleavage, the first series of a cell division by a mitosis after the fertilization. Cell division in a cleavage is rapid. New cells do not take a time for a growth phase, that is a G1 phase. Cell growth does not occur, so the cell decreases in size with each cleavage division. So these are the divisions. This is for a sea urchin and this is for a frog. 2 cell stage, 4 cell stage, and 8 cell stage. These are the 8 blastomeres. Here 4 blastomeres and 2 blastomeres. Early cleavage results in cells of a smaller size. The morula forms, it is a solid ball of the cell. Blastula form hollow balls of the cell. Here, after the fertilization by a sperm of an egg cell, zygote is formed known as the fertilization. Then the zygote becomes an embryo, 2 cell stage, 4 cell stage, morula, and the blastocysts and this blastocyst having a blastopore to a vegetal spore. In a cell differentiation, the first event, that is a glastulation, one of the side of a blastula get invaginated and forms a glastula, and in this glastulation 3 germ layers are formed, and these germ layers are known as a endoderm, mesoderm and ectoderm. The differentiation means the changing of unspecialized embryonic cell into a specialized cell, tissue and organs of multicellular or animal. The ectoderm, which is the outer layer of the blastocyst, and it gives rise to a nervous system including brain, spinal cord and nerves. The ectoderm gives rise to a lining of mouth, nostrils and anus, epidermis of skin, sweat glands and hairs, nails. Whereas the mesoderm, that is a middle layer, gives rise to a bones, muscles, blood vessels, reproductive and excretory system and inner layer of skin. Whereas the endoderm, innermost layer, it gives rise to a lining of digestive system, lining of trachea, bronchia and lungs, liver, pancreas, thyroid, parathyroid, thymus and urinary bladder. So these are the later stages of a fetal developments. So thank you for watching my video. If you like my video, please subscribe my channel Easy Life Science, share it to your friends and don't forget to press the bell icon so you'll get the notification of my newly uploaded video. So thank you once again.